data is going to be Jairam. And Jairam is going to talk about Epiviz, which is a tool for looking at epigenome visualization.
Hello?
Can
you hear me?
Can you hear Jairam?
Okay, all right,
Yep.
fine. I think there's a question in the chat. Someone wants to, can you bookmark a specific configuration to share?
Yeah, I think Sean sort of indirectly answered the
Oh,
question. I think they were talking about a specific gene. You can't quite yet do a specific gene, but you can do a profile and a view within that profile, but not a specific gene. But that's a good suggestion. We should take that back to the engineering team.
all right, sorry,
And Joshua just replied saying that, yeah.
Yeah, that can be hacked. Mm-hmm.
All right, I'm going to share my screen. I'm going to set it up. Give me 1 second so I set it up so I can see the chat and the messages. All right. Can everyone see my screen?
Yes, thank you. It
All
says
right.
Nemo.
yeah. So going back, we're going to continue to use the audience feedback that is available through Zoom. Please let me know if I'm going fast or slow, or if you have any questions, please post them in the chat. Actually, all right. So in the morning session, Brian talked about how to analyze and look at expression data within the Nemo Analytics platform. I'm going to focus more on visualization of epigenetic data. If you're starting from the raw reads, you'll probably use many of the pipelines that are available through the Terra platform to generate either signal files that are either coverage files, signal files, or you can also probably run through peak calling and generate peak files. What I'm going to focus on is the visualization of epigenetic data rather than the analysis and the pipeline side of things. And the first thing we all think of when we talk about visualization of epigenetic data is genome browsers. If you're not familiar with genome browsers, these are widely used tools for visualization. They take advantage of the linear organization of the data along the genome and then visualize data as tracks. In addition, they also provide many annotations along the genome, for example, location of genes, promoter regions, enhancer regions. And they also have functionality to quickly navigate to either a gene of interest or a genomic region of interest. So Epiviz is also a genome browser that we've been working at the University of Maryland. It is built in a modular way so that it is easy to integrate with applications, or you can also embed them on web pages. One of the unique features is in addition to supporting track-based visualizations, it also supports plot-based visualizations. So if you have a matrix-like data sets, for example, expression data, you can use heat maps or scatter plots, which is much more intuitive to look at than looking at expression as tracks. And some of the other features include like brushing, where you highlight a region in one of the track, and that immediately highlights that region across all the other tracks. So as part of the consortium, we've generated a couple of workspaces in the beginning for the flagship papers. So this one's coming from the motor cortex mouse MOP dataset. So the first one, the image you see on the left, shows you the different types of visualizations and also tracks that are available. So the first one is a gene annotation track that shows you the position of gene. Currently, this is in the GAC2 region, so you see the gene symbol. The blue bars represent the exon regions within the gene. So the second track next to it is, so this data set, they've identified clusters at two different resolutions. One is the major clusters. For example, the major clusters are glutamatergic, GABAergic, and then within each of these, they've identified a second level of subclusters within them. And those are the level 2 to 3, the p-val, and so on. So the second track you see here is showing you peaks. What we call on Epiviz is called as a blocks track, so that if you can quickly look at this and then say which cell types have peaks in this given region. As you notice, the L2 to 3, all of them have a peak in the beginning of the GAC2 region, but it goes to but only the GABAergic types have it later on. The next set of tracks is the signal tracks. This is looking at coverage across each of these cell types. And you notice a similar behavior as well for each of those minor clusters. And the last one is the methylation track. What Epiviz can also do is also do computations in the background directly over files. So for example, since we have clustering at two different levels, the major clusters and the minor clusters, you can also say on Epiviz, I want to aggregate data, take a mean of all the data of all the minor clusters and only show that result. And that's what you see on the right side, which is the same exact data, but in a different view. So this one's showing it's the same signal from the first three tracks here, but then we actually calculate a mean of it and then we visualize it. And these are not pre-calculated files. These are something that happens when the browser is initialized and when you look at the data within this region. And we generate a couple of workspaces for this. Second, I'm going to actually walk through this profile that is actually available on Nemo. So if everyone has linked to either the outline document, which I'm going to post in the chat. Okay, sure. There's a link to the epigenetic profile, and if you click on it, it should take you to the Nemo Analytics page. But then, if you see the profile, it should automatically fill in as the mouse MOP epigenetic data. If it does not, you can always click on it and then choose the mouse MOP epigenetic data. That's the third one in the list. And I hope everyone's at that point. We can search for a gene of interest, similar to what Brian was showing us this morning. If you type in if you know the exact gene symbol, you can do an exact match. If not, you can do the search and then it'll give you a result of all the matches. And once you click on search, once you click on search, it should show you the profile. So the first one is similar to what Brian showed this morning on how to take an expression data and then create a side-by-side t-SNE view. So this one is around the GAT2 region, where you're looking at expression across all the clusters that were found, cell types that were found.
You make this is the presentation mode, right away the screen. You click on the bottom, you can make that as a presentation mode. Yeah, you can click that's why make it bigger.
Okay, I see.
Okay, thanks.
All right.
Better.
Yep. So the first one is similar to what Brian showed this morning, where you're looking for a gene symbol, and it's going to plot that expression of gene across different cell types. And it also has an overall view of the cell types with their labels, what those are called in the t-SNE plot. The second panel you see is an epigenetic panel. And since we started this region at the GAT2 region, if you look at the gene symbol, at the middle of the screen track, you should see GAT2. There are a few navigation options that are available. You can either zoom in or zoom out of this region. You can also navigate left and right, so you can increase the region around this gene that you're looking at. The second set of tracks are the ATAC signal that is generated for this paper. And the first thing you notice is they're not using the same y-axis across all the tracks, and that's by choice. If you're trying to compare different signals across this in this visualization, you will want to have a fixed axis across all of them. So we have an option to do that. So if you see this icon on this visualization, that's the third one. and you click on it, it should automatically set the same y-axis across all those visualizations. Now you can make a comparison across all of these cell types and then figure out which ones actually have open chromatin in the region you're looking at. And a couple of other things on this is you can also change the colors of each of the tracks. For example, that's the color palette panel over there. If you want to change this color, for example, to a blue, and then this one to a red, for example. Actually, there's already a red. Let me choose orange. Well, that's already orange. Yellow. It immediately reflects those changes right away. And the next thing you can do is the cog icon. There's a cog icon that allows you to change settings. So on Epiviz, every type of visualization has its own set of settings that are associated with it, depending on what track you choose. So for example, if you use a heat map or a scatter plot, you can define clusterings. But for signal tracks, you can define binning parameters and other stuff, and I'll walk you through each of those. The first set of settings you can do is you can actually add a title for this visualization. So for example, ATAC across minor clusters, and then you'll see a title over there. I'm going to click on that again. This one I'm actually going to do on the second set of panel over here. So I'm going to do the same exact thing, which is realign all these data sets to use the same maximum max Y value. As you can see, the CG methylation tracks are very dense data sets there. So they have a lot of data points within this region. And that is good, but that may not always be good for visualization. So what you want to do sometimes is to be able to scale the number of data points down. So one of the parameters we provide here is what's called as a step parameter. And what that does is, if I choose 10, for example, it's going to take, for every 10 base pair region in this window, it's going to calculate an average of all the data points that are in that window. And then it's going to plot the data. So as you can see, once I change that to 10, it's now replotted the data using that averages. And you can choose a higher value, but bear in mind that as you increase the step parameter, you're going to lose the resolution of data that you look at. So an optimal step value is always something you want to play with. There's a couple of other things. So when you can also change the interpolation function, which is the smoothing function that you use to draw the line across the data points. By default, it's chosen, the basis is the function, but you can always choose linear, and then that should update the track. Actually, it might be linear on this visualization. But for example, if I choose step after, it's going to change. It's going to give you more of that histogram kind of a plot. And you can always change these and save them as defaults for any workspace that you create, or for any epigenetic data panel that you create. Coming back to the slides. So I walked you through the overall the window of Epiviz. One thing I forgot to mention is for every epigenetic data panel that is available on a profile, there's also a full view, so you can click on it and it'll take you. I think I already have it open somewhere. So you can click on it and then it opens the genome browser in a full window, and that's and so that you have more screen real estate to look at all the. If you have a lot of data that you're looking at, you have more screen real estate to look at all these tracks. But you can also navigate to any gene region of interest that you're currently interested in. So going back here. I'm going to quickly go over some of the features of Epiviz in case I forgot to mention it while I was showing the demo. So this is the fixed axis where you want to be able to make relative comparisons across different signal files or different cell types within a visualization. Another thing that Epiviz also provides is transformations that you can do. So for example, if you're looking at replicates, for example, you can use the mean as a transformation, and that only visualizes the mean of the signal across all the replicates. You can also do things like log2 ratios if you have two different groups of signal or cell types, or you can mirror the tracks against each other to make a more quick comparison of the signal. As I was showing, the Epiviz also provides the parameter step to do aggregations of data within the window that you're looking at. And that step parameter is important if you're looking at a data set that has a lot of data points, but you want to scale down the resolution a little bit. And that's available on all the line-based tracks that you see, or line-based visualizations you see. If you're using heat maps, clustering is a commonly used technique, which clusters. So if you're looking at a heat map, your rows are samples and your columns are genes. You can either cluster by samples, you can cluster by genes, or you can cluster by both of them. And there's different options that are available to choose what clustering metric you want to use, what distance function you want to use for the cluster, for generating the clusters. The other thing is, so if you have a lot of cell types, for example, and there's only a few of the cell types that you're interested in, you can also hide or show those individual tracks that appear on the visualization. And this is especially useful when you have 20 different cell types for a given data set, but you're only interested in one particular type of major clusters, for example. You can change colors for all of them. That's something I've shown during the demo. This is just a recap of the entire structure. So all the data that is available on NEMO archive, and once you run it through a standardized pipeline, either through Terra or if you run it locally, you have these big wigs and big bits that are generated for either the signal files, the coverage files, or the peak files. And once you have those, you can then visualize them using the inbuilt epigenetic viewer on NEMO Analytics. So I'm gonna show 3 steps in this process. So for example, first one is the upload interface. The upload interface is how you would add your epigenetic data into the NEMO platform, the NEMO Analytics platform. We're currently limiting I think the uploads are limited per user currently, but you can always send us an email to add, if you're trying to add a large data set or a collection, do send us an email and we'll be able to increase or increase the limits, the storage limits for you. So going back to the home page, on the top right corner, you see an upload epigenetic data set link, and it takes you to this page. So we support what's also called as a track hub configuration. So if you have a data set and you're already using the UCSC genome browser, NEMO Analytics can directly pull in all the information about the files and its metadata from that track hub configuration. The only requirement for this is this track hub has to be accessible publicly so that we can pull in the information. It cannot be within a VPN or a firewall, because that restricts the access. That restricts how NEMO Analytics can read those files. If you process them and generated big files locally on your machine or in your lab, you also have the option to individually upload all those files to NEMO Analytics. And for each of the file, you can specify a title, you can add annotations to them. And since these are files that you generated, you also have to mention what reference genome was used to map those reads and to generate the files. We currently support most genomes, for example, the MM10 genome, 2 different versions of the human genome, the HE19 and the HE38, and the marmoset genomes. If there's a data set that you're uploading and the genome is not available, let us know and we'll be able to add that in. And for each of the files you upload, you can also set the access levels. If it's public, anyone on the platform will be able to access the dataset. But if you choose private, only you will be able to access the dataset and then create visualizations and profiles. And once you're done uploading all of them, if you submit it, they get added to the interface, to the application itself. The next step, once you upload the epigenetic data, is to actually create the visualizations from that epigenetic data. So that is done through, if you go back to the top right corner where you click on your name, there's a link that says Epiviz Panel Designer. And if you click on it, it should take you to this interface. And the first thing you can specify is provide details about the panel itself. So if this is a collection of data that is going with the publication, you want to add a title, add the description of what the data sets are. You may also want to explain how they were processed, and then any information that the user can get by looking at the description. You can also set the panel access, if it's public or a private. A public panel, just like how Brian was showing before, any user can add this panel to their own profiles. But if it's a private panel, only you will be able to see them and only you have access to it. And as always, only you will be able to access it. The next is the genome build. So given the collection of data that you're looking at, choosing a genome build will automatically update the annotation track. But for the purpose of the demo, I know that I'm going to choose some of the mouse data sets. So let's choose MM10. And we are in this region, which is the PND 521. The location of the region itself does not matter here, because when you add this panel to a profile and you search for a gene and go to that location, the epigenetic panel is automatically mapped to the gene you search for. The first thing we're going to do is click on Add a Chart, and that should pull up all the publicly available data sets that are currently available to everyone. So a few things here to walk you to this interface. There are a lot of visualizations that you can choose from, and our manual has documented on how these look like and for what type of data sets you want to use. So quickly going through, the blocks tracks are the ones you would use for peaks. Any files that you run it through a pipeline would call for peaks. Heat maps and scatter plots usually for gene expression data or matrix-like data sets, and the rest of them for signal or coverage files. For today, we're going to choose the multi-stacked line track, and then I'm going to choose the first 3 data sets here, and then I'm going to click on Add Chart. And that would add a visualization to the Epiviz panel here. And as I said, when the visualization starts, every track gets its own y-axis. So you can set that by clicking on this icon. And now you choose the same y-axis across all the files that you've chosen. You can also update the colors using the palette icon here. Or you can specify, add any other settings. So. I can also change the step parameter. I can also choose what interpolation technique I want to use. Since I chose a step parameter, it's not this data set is already sparse. The step parameter makes it even more sparser. All right. And once you make all your changes, you want to click on Save Epiviz Panel. And once you click on that, it won't let you make any more changes to the panel itself. You won't be able to add any more data sets. We're currently working on an interface where you can, once you create an Epiviz panel, you can go back and edit the panel and make more changes. But for now, it's gonna blurry out the screen so that you won't be able to make any more of those changes. And hopefully everyone's there at that screen. As Brian was showing before, you can then go to a profile. Since I'm running out of time, you can go to a profile and then look for this panel and then add this panel to a profile and look at data. And does anyone have any questions?
Jerome, there's a question in the chat.
Yeah, I'm looking at. Can we add external annotation tracks? Yes. So through the upload interface, as long as you know where that external file is stored, you can add that external file and then you can create tracks based on that. Yep.
All right. If there are no other questions, I'd like to thank Jairam for the presentation and walking us through the demo. Michelle, do you want to stop and restart the recording for the next session?
Okay. I can find the right button.
Yeah, so we had to do a little